what I was tasked with for the next 20 or 25 minutes was just to give a broad overview of what is somewhat vaguely been called modeling challenges. Yes.
I just object to
Yes.
this being tasked phrase. So we decided together that we
We all decided together that this is something that I will do.
Same here. We all decided that I'm supposed to do the
Okay, and so I suppose it's up to me to interpret what we mean by modeling challenges, or it was up to all of us. And so I really just want, I mean, this could have become a lecture about here are the 20 different kinds of modeling approaches that people have tried. Here's what's good about them. Here's what's bad. I think that is not something that I'm going to be able to do. So I'm really going to endeavor to make three, I think, relatively simple points that maybe many of you already know, but I think hope set the kind of stage for what I believe should maybe be the modeling arm of this kind of meeting. And those points are basically quite simple. The first one is the sort of traditional kind of models in ecology that we kind of think about. In particular, maybe many of us here think about what are called Lotka-Volterra models, which are shortened as GLV. And some others think about what are called consumer resource models, which are labeled CRMs. They traditionally neglect energetics or thermodynamics. I think this is somewhat uncontroversial, but I'll hope to make this point clearer over the next few minutes, what exactly I mean by this kind of neglect. I'll then hope to kind of point out what's the key kind of conceptual bit that is kind of missing from these models. Conceptual bit that's missing. And I think that's really, there's kind of two ideas. I think one of them Orkun brought up before, which is we often, especially, I mean, in a lot of Volterra models, usually we're only thinking about species interacting with each other directly. We're not really thinking about the resource and environment. But even when we're thinking about species interacting through resources, we're often thinking as resources as one entity, nutrients as one entity. And I think one thing that sort of this redox framing kind of teaches us that when it comes to thinking about energetics, about microbes, especially harnessing energy from their environments, it's really two nutrients that you need to think of, a donor and an acceptor of electrons, primarily because getting energy is a process of moving electrons between two things. Yeah, so it's really, I think the way that I kind of think about it and the way that made it clear for me was it's really two nutrients, not one. And again, I'll make it clearer sort of what I mean by this. And I think the sort of consequences for ecology are not just that this is an additional complication that we need to put in our models. That's not kind of my goal. I think the kind of central thing that that the models miss by not including something like this is they kind of miss the sort of dynamic feedback between sort of rates and yields of processes. Okay, so I'll also hope to explain the essence of what I mean by this. And then finally, I'll hope to talk a little bit about some simple ways of including this kind of including this kind of feedback through this kind of idea of redox. And then I'll hope to make the point that we are kind of far from kind of getting this right, and hopefully this meeting is kind of serves to be a catalyst for this. Okay. Okay, so
can I interrupt here? This whole thing about two nutrients, how is it different from, say, organisms needing carbon and nitrogen? And they are also two nutrients, so they are also connected to some law.
Yeah, great point, and I'll just use this opportunity to say, please feel free to keep interrupting me through the talk. At some point, if I look at the clock enough, I might ask you to pause, and it'll be okay. About this whole two nutrients business with carbon and nitrogen, it's totally true. Like I said, the talk today is kind of focused on energetics. When you think about, you could also think about this kind of dual role of nutrients, like microbes in principle need nutrients to do two things, to get more matter as well as to get energy. When it comes to getting matter, of course, you need all of the elements in sort of principle. I think there are similarities between having these two or three different things, but also some kind of differences. So when I come to sort of the energetic part of two sort of nutrients, I'll point out what are the differences and what are the similarities. In particular, I think this kind of dynamic feedback between rates and yields is kind of one of them.
And again, to me, the real difference is that when you start thinking about thermodynamics, it's really universal, whereas whatever you see in ecology is totally idiosyncratic. You know, one micro, not at all like that.
I thought that sentence was going to go a different way. Okay. Okay. Thermodynamics is universal. I think we'll all agree to that. But I think my understanding of what I generally hear from people when speaking about this is how much it matters to actually, affect the dynamics or for our ability to understand what's going on or to make predictions. That I think my understanding is your mileage may vary. And that's kind of one of the things that I hope to kind of make clear towards the end, like, where are we right now? What are the situations in which we really have to care versus not care about this kind of complication? To me, is not entirely clear. There are certainly contexts in which it matters a lot. I think the field of anaerobic digesters, etc., kind of showed us great successes where these details really matter. But I think to me, a big question is how much do they matter in sort of other diverse microbial contexts? To me, the answer is not clear. There were some other hands. Yes.
It's not a clarification. So
Yeah.
it's the assumption that a redox couple is always needed to extract energy.
Absolutely.
I'm thinking in terms of, let's say, anaerobic glycolysis, where the NADH that you burn on you also get it back. Should that be also considered?
My understanding is yes. I'm looking to some other people in the room because they're more experts on this than me. But I think in the case that you're describing, you still need a donor and an acceptor, except I think in this case, you kind of generate one of them internally. Is that roughly right? Yeah. Okay, I'm getting the thumbs up. Someone who knows more than me. Yes.
Just to follow on Sergei's point, I wanted to suggest that there's an opportunity to take back the term ecological stoichiometry, which was such a promising term, but got unpacked to such a disappointing level, right? So not only one or two, but not infinitely fine-grained, everything is idiosyncratic. We sort of understand within ecological plasticity that there are groups of things that act together. And if we want to couple ecology to evolution, it seems like there should be a way of finding out what a good coarse graining level is for a given kind of substitution question or something. And all of that is in principle within reach. So I wanted to ask if that's within the remit of where you see going.
That is within the remit of where I see it going. That's not within the remit of where I see the stock going, because the stock is specifically about energetics, because that's what we all we would talk about. But yes, I think in general, the idea, this kind of idea of universality of thermodynamics and universality of course, needing the elements to be able to grow and make more of yourself. I think these ideas definitely have more, they need to be put into conversation with each other. At the risk of not going beyond my three main intro points, maybe I will start, but this is not to discourage you from interrupting. Yeah. Okay. All right. So I won't sort of belabor the point. Some of you here probably already know about these Lotka-Volterra models. My goal is not to tell you what they are, but just the kind of idea that I want to pose to you is there are different modeling approaches in ecology. The sort of maybe simplest one that you might say is this idea of using Lotka-Volterra models, where really in some sense, you're only thinking about the species and the way that species affect each other's growth. And the way that you do it is through some interactions. So the key idea in Dr. Volterra models often is that the interactions between species or strains or what sort of have you are fixed. They don't change with the environment, even though they could in principle. But there's no systematic way. The model kind of tells you how to probe that. Okay, there is a way to do that by sort of thinking about where the interactions come from. In many of those cases, those interactions can come from the fact that microbes are competing and cooperating with each other for different nutrients. And so you might think of that in a more mechanistic form through what are called consumer resource models. And so usually the way that we're thinking about consumer resource models, so here I'll just kind of try to draw a comparison between what I'll call conventional CRMs, as I understand them, with maybe what I'll write down is the redox view that I've learned about in the past little bit, is sort of the following. So, I mean, the sort of standard way you'll see kind of diagrams about consumer resource models is that you'll have a bunch of species and you'll have a bunch of different resources. And, you kind of prescribe which species can eat which of these resources with what kind of consumption preferences. And that's essentially your way of sort of modeling metabolism. And there have been some people who say, okay, you can think about energy in this way, in these kinds of models. And the way that you kind of think about it is that you think about essentially all of these different resources. So that would be the triangle, the square, and the circle on some sort of energy axis, on some sort of axis of energy content. And that's something that may allow you to say, okay, this is a more juicy resource, this is a less juicy resource, and so on and so forth.
You also include crossfeed in here, so arrows go both ways, right?
Good, yeah, great point. I sort of deliberately didn't draw arrows over here just to kind of point out that microbes can consume nutrients that are provided from the environment and also produce others. And so this can become the basis of sort of this competition and cooperation that I was kind of telling you about. And now you can think about interactions between these different species as sort of emergent consequences of this process of competition and cooperation through resources. Okay, and so the way that you're kind of thinking about metabolism or energetics here is you think of a set of consumption preferences for every microbe. And then you think about a growth rate G sub I of species I as just some nice little sum that goes something like this. Basically, the only thing that I want to kind of point out is the following. These R sub alphas are sort of the concentrations or the abundances of all of these different resources at the present moment. So you might want to talk about it in time. And these CI alphas are kind of what is sort of schematized by all of these arrows over here. These are kind of consumption preferences that kind of tell you which species like to eat which resources. And what you're really thinking about is that the growth rate of any species or strain is just going to be a nice sum of all these CI alphas times R alphas. And so this is the thing that I want to kind of highlight here is you're really thinking about a situation where every resource contributes to growth independently and additively, right? This is, of course, a simplifying assumption, and in many cases is not true. And this is kind of exactly what the redox energetics or the redox metabolism view kind of rails against. Okay, so in the redox metabolism view, the picture does not quite look like this. The picture looks a little bit like this, and for that, I won't draw this entire thing again. I just want to zoom into one of these microbes. And so what this picture is trying to say is the following. This is a slightly more ovular shape of what Orkun drew this morning. This to me is definitely a cell. And so really what you're kind of thinking about is to get energy, what this cell has to do, and I'm going to kind of repeat what Orkun said, so I'll go a little bit fast, is that you need two resources. Okay, and so, I often make models, so I would represent these as what I'll call R1 and O1 and R2 and O2. but I'll give you some sort of names to give you a picture of what really we mean here. So for instance, all of us over here are doing the same process to get kind of energy. What we usually do is that we use some form of organic carbon like glucose, couple it with oxygen, which conveniently is O2. And this glucose we respire out as carbon dioxide. This oxygen becomes water. You know, this is just an example. This is something that's familiar to us, that's taught to all of us. And really what's happening in the middle that's kind of connecting these two yellow arrows is the process of moving electrons from sort of one arm to the other. And it's really this process of moving electrons is the way by which all of us, including microbes, get sort of energy. There's and so this is sort of why the idea of a donor and an acceptor comes in. This bit over here, you know, you can think of glucose or organic carbon as a donor of electrons. Just look at the arrow of the direction of electron flow, and this oxygen becomes the acceptor of electrons. Right? So you kind of always need a place where some high energy electrons come from, and a place that where they kind of end up at, and in that difference kind of sits your ability to extract energy. And it's that energy difference that you can then harness for all sorts of things. Cells will often couple it to some energy currency molecule like ATP and so on and so forth. But that all those nitty-gritties I don't want to go into. I'll take a question now. Yeah.
Yeah, a very kind of high-level point here. So you kind of contrasted the conventional CRM being linear and redox being nonlinear. I think this is the wrong comparison because there are plenty of CRMs which are nonlinear. Take the Liebig's law of the minimum or plug in Monod's grow, and it will be CRM. What really differs here, if you want to just a high-level zoom out, is that in a regular CRM, your byproducts don't affect your growth rate. You excreted them and forgot about them.
Yeah.
In redox view, as well as in any kind of place where you have the energetics, you have this product inhibition, which is
you're absolutely right. Yeah, you're absolutely right. My aim, maybe I shouldn't have made the sum here because that was somewhat confusing. My aim really wasn't to point out the fact that you sum over and have this linear term. In fact, you can have many other things. My aim indeed is going to be to show that what we think of as Ci alpha here, which is a number, is actually not a number, but actually something that responds to the state of the environment.
State of byproducts. So
state
it's very
of the
difficult to separate this sort of what is substrate and what is a product.
You're absolutely right. I will make this point. Yeah. Okay, great. And so in this picture, this whole tower of energy content is not exactly true. What is instead true is sort of the following. You still make a tower, but what you make a tower of is some sort of potential, or in this case, chemical potential. And you can essentially organize all of the different, what I call half reactions, which are just these R1O1, R2O2, R3 O3 pairs on this tower based on their intrinsic ability to donate or accept electrons. You choose a convention. I don't remember now what this convention is, but this doesn't quite matter. I'm just going to put in abstractly R1O1, R2O2, R3O3 here. And really the idea in this picture is not just, is very different from this picture. In this picture, you can just have one nutrient in principle, just the triangle, and be able to grow just fine. In this picture, that's not true. And so in this picture, the way that you get energy, especially if your growth is limited by energy, is that you couple one of the arms on top with one of the arms at the bottom. So this is, for instance, one way of making a viable bug. This is another way of making a viable bug. And this is indeed another way of making a viable bug. So you always start up and you always go down. Yes?
Would you like to make two towers? You can couple different things. Like what you could make a redox tower and couple different reactions. So you could actually have two
Do you mean to say that I could have one donor but two acceptors?
Yeah, no, you can couple different redox couples to show that microbes actually are extremely diverse and they can pick depending. So it's not just so you
Oh, I see.
you can couple the redox couple glucose to carbon dioxide with not necessarily oxygen and
Good.
water.
Yeah. So if I understood correctly, you're talking about the plasticity or flexibility of the same microbial cell or species to be able to, in some conditions, be able to couple this with that, in some other conditions, be able to couple this with that, and so on and so forth, based on what's available. That's absolutely correct.
Let me ask another question. So in
Yeah.
your framework, so Li and Lj can coincide, or I mean,
Sorry, I just want to clarify. This is not my framework. This is, as far as I understand, the framework. Yeah.
I mean, so I'm somewhat confusing about how this pair Li and Oi can be determined, and so in this
Okay,
framework,
I see.
Li and Lj or
Okay.
Oi and Oj can coincide.
I see. So the question is, how do you figure out these pairs, right? You do somehow, I don't know, like chemists know the pairs go. Someone help me out here. As far as I know, they're given to you by.
As of
My God.
Yeah.
I mean, that's you.
You do the redox potential
Well, I mean,
for
like
equilibrium
chain
transformation, and then high redox potential to lower redox potential creates the difference.
No, but I guess the question is, how do you figure out
so
that
yeah.
glucose goes with carbon dioxide and something else goes with something else? And I don't know. Someone clearly knows the answer to this question. I do not.
You write the delta G, the given the end.
Of course, I can understand.
Yeah. Okay.
Wait, how do you figure out like experimentally or intellectually?
So what's the question?
Intellectually. Okay.
Intellectually.
Intellectually.
C13 labeling.
Yeah,
I mean,
labeling experiments.
electrochemistry is what this is about, and people have been trying to figure out which compounds act as redox carriers.
Okay.
So we have to go buy some books and read about that. But I guess some of these redox pairs are well understood, but some of them are still being discovered, although Eric wants to.
It's fine. You just have two different discussions and you're using the same notation for them. There's an elementary redox couple that happens when the electron carrying state of something changes. That's the thing that Sergey's talking about. And you can graph that by midpoint potential, and that's defined. You know how many electrons change, and then you know what the exchange potential is for that. Then there are all these composite things where there's a bunch of reactions that convert glucose into CO2, and you can shorthand that with this kind of a diagram, but it doesn't mean the same thing. So, and both of them are alive in the discussion.
Okay. Maybe one last question before I try to make some more points. Yeah.
It's just like, so I'm a biologist, I have a naive question. So like still this GLB or CRM,
Yes.
they're still like, good to answer certain questions. If you put like another dimension to it,
Yeah.
then it requires like more experimental data. And I always question like, having that, how much improvement we can have in a particular like presumption, because nothing will be 100%.
Yeah.
But how much improvement, and as a biologist,
Yeah.
is that worth enough to me
Yeah,
to get
no,
into?
I think that's an excellent question. And indeed, my aim here is not to poo-poo on both of these classes of models. I am equally guilty of sort of working with both of them. It's not because there's like models are bad or models are good. It's just because what's the question, right? So if I get there in the next few minutes, I will hopefully show to you one simple example in which it actually matters, in which actually the energetics matters, and both these frameworks would get the answer wrong. And in this case, the answer will be about diversity. Okay, like how do you get things to coexist?
Just have one comment. I think your writing is too good.
My
If I write something in the board, like I cannot understand it. So like everything is so good.
Yeah.
Okay, thanks. Okay, that's great. Okay, so I was just telling you guys about my understanding of what I think, how I come to understand the redox tower. Yeah, but really the point that I want to kind of make is what you think about here is a set of consumption preferences becomes here in this picture sets of donors and acceptors that you could in principle couple to. right? So that's kind of, in some sense, if you think about energetics, that's kind of the right thing to kind of think about. And then the second point that I want to make is that in this picture on the left, this I think Sergey will agree with me. You're essentially thinking about irreversible processes, right? You're thinking about taking in a nutrient irreversibly, converting it into microbial biomass. That's a simplifying assumption that here things are irreversible. But here, because on the right, because this is fundamentally a thermodynamic process, things can in principle be reversible. And this is indeed something that really, really matters, because this is where the connection between thermodynamics and kinetics comes in. Okay, and in the interest of time, I mean, so technically on the schedule, we have this happening until 11:20, which finishes in 4 minutes. We have a 10-minute phantom gap, at which point at 11:30, we're supposed to have icebreaker. Maybe if it's okay with people, we can go till 11:25 and then break. Okay, but yeah, if at any point you want to stop, just let me know. Okay. Okay, so this is kind of most I wanted to say about what the kind of central difference is between the sort of conventional CRMs and sort of the redox view. But of course, we're not here just to talk or learn about energetics. We're here to learn about energetics in the context of ecology. And so in my view, the kind of big consequence of this view on the right for ecology is really the dynamic feedback between the rates of processes and the yields of processes. So let me kind of describe what I mean by that.
You can pull you can save time by pull the board.
I can save time by doing what?
Pulling the board. I
Oh, I can
use it. There's a board.
Oh,
Yeah.
I didn't know that. Thanks.
2 seconds.
Yeah, that's great. I didn't know it went that way. Okay, so the main consequence for ecology it's really that the rates of nutrient consumption energetic yields they're sort of dynamic and they're an emergent consequence of the feedback between thermodynamics and kinetics. And let me kind of describe here what I mean. So what is really happening here in this picture on the right is whatever I've labeled on this tower as chemical potential, this is not one number specifically. This is a number that's very sensitive to, like Sergey was saying, the concentrations of the different nutrients or these kinds of molecules that are available. And so in principle, as you consume more and more glucose, produce more and more carbon dioxide, consume more and more oxygen, produce more and more water, you in principle change these chemical potentials. And so what can happen as a result of that are two things together. First of all, the energy difference between these two things changes. And so that affects the yield of the reaction and the sort of energy yield by which you're able to, that you can extract. And in principle, as the sort of general rule or the general kind of thing to think about is as you accumulate more and more product, you kind of lower the chemical potential difference between these two couples further and further, you get a lower and lower yield. And at the same time, because of the intrinsic connection between thermodynamics and kinetics, you also lower the rate of the reaction, right? So in some sense, if you're thinking about it in the CRM picture, what you think about, this is kind of one pithy way that I understand it. What you think about is these CI alphas for any species I using nutrient alpha is actually secretly a function of, not all, but at least some of the concentrations of these OIs and RIs. This is kind of what I mean, sort of not fixed, but dynamic. And again, I don't want to give you a whole lecture about how you might want to model some of these things. I'll give you one sense of that tomorrow in my talk. But I just want to take maybe a couple of minutes to tell you one nice and simple example that I learned of from one of Orkun's papers, which I think really kind of encapsulates what I think I'm kind of trying to talk about. And this kind of also goes into simple ways of including feedback. So really what we discussed so far is we're thinking about this kind of net reaction. In that case, R1 plus O2 going with R2 and O1. And what we also learned is that this fundamentally is a reaction that's reversible. So in principle, it has a forward flux and it has a reverse flux. And in principle, of course, what you care about is the net rate of this reaction. And that, of course, is the forward flux minus the reverse flux. And there's some ways to kind of derive and show that you can kind of write this down as the forward flux times essentially some thermodynamic factor. Maybe I should be careful in writing all my KTs or RTs and things like that, but that won't be so important to the point that I want to kind of make. The point that I want to make is the following. The net rate that you kind of get, this would be kind of the analog of what I would call CI alpha. Kind of now has these two parts. The first part is the rate of the forward reaction. This is kind of what you would model in a conventional consumer resource model. So this could be something like, there's a maximum rate, and then it depends on the concentration of what you're using. Maybe there's some sort of monoform to it, just like Sergey was saying, it doesn't have to be linear, right? The key point is this whole thing sitting over here. This is essentially your thermodynamic drive factor. And this is kind of the key thing that's controlling what I'm calling this feedback between thermodynamics and kinetics. The idea is kind of simple. You can have this delta mu over here is kind of what I meant by this chemical potential. That's sort of what I label by mu. And so delta mu would be some analog of this sort of gap that I was talking about. So think about a particular organism coupling this to that. The idea is when this gap is very large, then of course you can kind of ignore this factor. This factor is essentially 1. And indeed, in some sense, I would say maybe conventional CRMs are assuming this, though of course there's no kind of concrete sense in which you might say that that's the case. But even though you might start off with this factor essentially being 1, as you sort of proceed and start to grow and you accumulate more and more product, just like Sergey was saying, this gap starts to shrink. And this factor starts to become more, less and less negligible. And eventually there will come a point at which this factor will essentially be close to 1 because this delta mu will become close to 0, right? And at that point, no matter how large your forward flux going on the right was, no matter how much of this stuff of your substrate you had to sort of begin with, your net rate will come grinding to a halt, right? And this is the essential kind of feedback that I'm talking about, does not really exist in CRMs. Now, you might say there are many reasons why it doesn't exist. And indeed, there might be many cases in which it might be sensible for it to not exist. For instance, you can many times imagine what are called open ecosystems. There are cases in which nutrients are constantly flowing in and constantly flowing out so that you're never really accumulating these kinds of products. That would be well and fine. That immediately lets you now think about a case of a closed ecosystem where, that would certainly not sort of be the case. But I mean, I would say generally, this sort of correspondence between when these things ought to work fine and when these things ought to work not fine depends not just, of course, on the question that we are thinking about, but also the context in which we're kind of thinking about. I'll take a few questions and then maybe I'll stop. Maybe we'll start with Purushottam and then sort of fan out like that.
I'm somewhat confused about,
Okay.
especially the nonlinearity, because I thought to get to this form, you have to kind of assume mass action. So the J forward is kind of constrained to be linear.
Yeah, there are kind of lots of ways to derive this. In principle, you don't like.
Monokinetics is not real chemical kinetics. That is just some.
Yeah, maybe I misspoke when I said mono. Maybe you should think of it more carefully as some sort of Michaelis-Menten form. There's all these ways of kind of thinking about what's going on behind the hood of how you write down this J forward. The universe of 10, 20 different models that I was talking about indeed make specific choices as to how you want to write this down. And so that's kind of not my focus here. My focus here was really on this other term. But yes, if you're interested in more of the nitty-gritty of how you get it, I'm happy to talk about it later. Karna?
Thanks so much for this, Akshit. I'm curious about your take about the prevalence of systems where you have a thermodynamic factor, driving factor, which is close to zero. I mean, on one hand, you have like aerobic oxidation or respiration of glucose, which is a situation where that factor is close to one. And then there's like classical syntropies where you're kind of close to zero.
Correct.
Where are different systems sitting? And like, can you give some examples where this is really important to think about?
Okay. I don't feel up to the task of answering this question, but I'll still try. I think the sort of general point that you brought up of trying to classify systems and trying to place them along this axis of where the thermodynamic drive is large versus small is, I think, a good exercise, and maybe, to my knowledge, is something that we as a community have yet to do for many of the systems that we kind of work with. That's something that I would potentially be interested in sort of talking about. That being said, to think about the two extremes, and indeed, I would imagine these kinds of aerobic oxidation type systems as being on this one end where you don't need to think about it. Maybe like methanogenesis or things that are happening in anaerobic digesters is maybe on the other end. Again, there are people in this room who've done way more work about this, so please feel free to chime in, but that would be sort of my kind of naive guess. Maybe we'll continue to go like this. Yeah, Pablo.
Hi, actually, I just had a slightly technical question. So when you derive this bracket.
I didn't derive it. I just kind of bequeathed it from.
I imagine.
Yeah.
You're doing J plus over J minus equals exponential minus delta mu. So I know that to be true for elementary chemical reactions. Here you have a microbial growth. Right?
That's right.
So which is very coarse grained.
That's right.
So is there a set of specific assumptions?
Absolutely.
For this to be true? Because I mean, to say that a microbe is just an organic elemental reaction is
no, you're absolutely right. There is a very specific set of assumptions that goes into kind of deriving this or equations of this type. I think these are also what are called Haldane's. I think like a version of this is what is called Haldane's equation. Actually, at some point, this other workshop last year that Orkun and I were, and Wenying and others were part of, Orkun had done the Herculean task of kind of creating this slide deck with all of the set of assumptions that go into deriving all of the different functional forms. So I'm happy to talk about more of those assumptions there. But yes, coming with every form of this kind is a plethora of assumptions that go in as to how you coarse grain microbial growth. Yes, Casey.
I just want to throw this out there maybe as a provocative idea, but like Karna's question was very interesting. I would have felt even less capable of answering it, but my good friend ChatGPT just gave me a very good answer to the question.
That's great.
And so I wonder if, like, for some of these discussions, it's actually worth just, sort of doing a little collaboration with language
Oh,
models.
but someone's got to do the fact checking.
Definitely,
Yeah.
but that's why we can fact check it as a community.
Yeah, but it's actually true. Like that's probably less likely to spew something wrong than I am. Yes.
I just want to continue this thing. I mean, well, there is some feedback, who cares? There are plenty of feedbacks.
Good.
You produce acetate,
Yes.
which is acidic,
Good.
it
Yeah.
raises pH,
Yeah.
your cell becomes less stable. It's a feedback.
Yeah.
I think that what is really unique about this feedback is coming back to this topic of universality I brought up before, is that this one is given to us by gods, whereas J forward
this
is
is
a
up-
mess. I mean,
to
this
us,
is
yeah.
microbe to microbes, genus to genus, whatever. It's
Yeah.
even strain to strain variable.
Yeah.
So that's why we should pay attention to this one. Otherwise, it's just, another feedback, and we have plenty of stuff in the model.
Yeah. And actually, I thought initially you were going to say, okay, there's a feedback, who cares for ecology? And actually, I was going to say, in a version of this talk that didn't involve as many questions and involved better time management, I was going to say that one of the consequences of this feedback can be that two things or two species that you originally thought were not going to be able to coexist, let's say this one and that one, can actually coexist now, precisely because as they start to do their things, you can end up with, because of this feedback, this situation where it's steady state, they're kind of extracting the exact same energy gap. And so that's in fact something that Orkun showed very nicely in one of his papers. It's ISME 2016, I believe. And so you can get more diversity than you would naively expect simply because of this feedback and not because of any.
Oh, you just need diversity. You just need to properly count all the molecular species. And in this reaction, there are 4 of them. So in principle,
But that,
you can have
but that just gives you a bound, right?
Yeah.
So you're just saying you can have those species, but in this case, you can show that they can actually coexist because of this mechanism.
And this is a good discussion for tomorrow after you and I will, and many others will.
Okay. Yes. I just want to be cognizant. I mean, I'm happy to take questions. It's 11:33 on this clock. So, Orkun, you can decide what we want to do, but I'm happy to continue taking questions if that's good. Okay. Yeah. Oh dear.
Or someone else.
You
No, dear.
don't ask me about.
No, I'm not asking. I wanted to sort of make a comment because I know that there's a lot of people in here. Normally these meetings don't expand beyond microbial physiology and ecology, but there's a number of people here this time from the earth sciences where it's common to think about much longer timescales than we're used to thinking about in this community. And so I want to reiterate a point that you made, which is I think often in the community that you and I and several other people in the room inhabit, we're sort of doing a dialogue between Lotka-Volterra models and CRMs, something like that. And because of that dialogue, we're fixated on the dynamics of the species, and the things that we can measure also fixate us on the dynamics of the species. And in the earth sciences and in microbial physiology more generally, we're often interested in processes that occur even when the species have no dynamics, right? So the limits of very, very low or even no growth or just absolute eking out survival. And it's not that CRMs can't treat that case, it's that we typically do not treat that case. And this kind of redox view that you showed us very nicely focuses our attention on the fact that we don't treat that case. And I hope some of the talks later today and tomorrow and some of the other days will remind us that there's lots of interesting stuff that happens in the world that isn't coupled to growth.
Yeah, that's a nice point. And also the way that I think about it, it's mostly that in ecology, we're often thinking about the species, just like you were saying, we're much more interested in the dynamics of the species, their ability to coexist, as opposed to the processes themselves, right? And my understanding is in the earth sciences, people are more often thinking about the carbon cycle or, something else that is somewhat at a different level of description than the species themselves.
Not exactly a question for you, but that bubble you drew, the arrows, because of the redox model and the redox view, does that have a name to it?
Does that have an aim to it?
Not like a name, or it's
Does the arrows have a name? Is that the question?
The whole thing, the chemical potential.
This, I believe, is called a half reaction.
No, the whole thing, the view of it.
That's a microbe. I didn't mean, so other than redox metabolism,
I think
Yeah, I
he's
guess you
talking
can call
about
that, yeah, because
catabolism.
that's
you can call it catabolism is what Alfred says.
catabolism.
No, it doesn't have a name. There's no name.
I just want to say that I think you've done an amazing job here articulating two asymptotics of the theory problem. And what I really want you to solve for me is the middle. The asymptotics are the energy on one side. So in this J
Yes.
reaction here, what you've told me is that if all the system cares about is energy, then the delta mu's matter, blah, blah, blah.
Absolutely.
And the CRM case is if all I care about is resources. But in fact, as you started by saying, growth is both the allocation of resources and the energetics to synthesize those resources.
Yeah.
And there are beautiful examples that I'm aware of in the nitrogen cycle, and surely people who know more about this, like Avi or Alfred or something, can articulate even more examples where the thermodynamics are not the whole story.
Absolutely.
By a long shot. And so for some reason, we're observing these phenomena where resources are dictating certain fluxes. And boy, we would love to understand why. And I think even a language is still missing at the level of the community.
No, yeah, I completely agree. I had here, if I had for lack of time, a set of challenges that I had written down. And at the top of that list was engaging carefully with the dual role of nutrients, as I was saying, this role of nutrients as both sources of material and sources of energy, and kind of having a framework that kind of marries both of them is a challenge.
But I think what I really want is a way to write that down as a toy model that looks like a CRM so I can intuit about it and isn't an FBA model.
Yeah.
It's too much.
Yeah.
Yeah, I would love that.
Wouldn't you say that one of them is always limiting, right? I mean, for a given microbe, you could put a mean function and put here the.
But I think
maybe
maybe Sepp is talking
by
about a
the-
case in which kind of, again, there's something emergent about, you could have cases where the same microbe was kind of dealing with the material limitation, and in some other cases dealing with
And
the
then you
energetic
switch from
limitation,
one regime to another.
and then also be able to sit somewhere in the middle, right, where it's actually dealing with I don't know, some sort of co-limitation.
Base of measure 0.
I don't think so. Wrong side.
Yeah, I think
For a given
sorry,
microbe,
Dan,
not
go on.
for-
This is quite beautiful, but I think the way to resolve at least some of what Avi talked about and some others is that unfortunately, this J forward is kind of where a lot of the action resides.
Okay,
And, so I realize that it's not what you're really talking about here, but it's not just a mess. It's a challenge.
Yeah.
And so, I mean, I would say that the Michaelis-Menten form you wrote down is kind of like saying, okay, it's just glucose out there and we find it because it's there. It's not that simple.
Yeah, no, I completely agree.
And it's not just a pure mess either.
And meaning that, of course, there's a possibility of doing science and making
Yeah.
some sense of it. Yes. Yes, of course. Yeah.
Just to add on to what Seppo was saying, I think, part of both of these views are missing is the dynamic view of the phenotype and the physiology, right? So both of these are assuming there's some fixed parameters associated with the organism. And in the redox view, the environment is changing what the physiology is. But there's this other layer, which is like, okay, so what determines whether an organism will consume one or the other or both at the same time? What are the physiology?
You're absolutely right. Yeah. Yeah, which is what determines what you're actually at any particular point going to couple with what else, given what's available to you. Yes.
Not so much a question, but there are two comments here that I think add important nuance to add to your theory. So one is expand the number of nutrients, because so you said 2 nutrients, electron acceptor, electron donor. Of course, this is fairly trivial comment. Ecologists would know that you can add a third one, which is like nitrogen or phosphorus, right? Your growth can be limited by carbon.
You're right.
Or it could be limited by a building block of biomass, right? So in the environment that becomes important. So at some point, all of the picture becomes irrelevant because a simpler limitation is at play, right? The second one is a more fine nuance on your exponential factor there. When you, as you said, we go to longer timescales and low energy environments, deep subsurface, that kind of thing. When you have very little energy, quantization of processes that's related to ATP generation starts playing a role because you cannot generate less than 1 ATP by a cell. And so at some point you have a sharp cutoff where cell metabolism stops, or rather you can have a switch in the cell metabolism. You go from active growth to maintenance. And if you further restrict the energy flow, you go to survival mode. And those modes seem to be quantized. There seems to be an order of magnitude difference, sharp drop between each of them, which is somehow related to the biosynthesis chain within the cell. And so
Are you talking about a specific example of a specific microbe, or?
It's been shown in specific examples, but it's also been shown across the communities.
I see. Okay.
Yeah. And so if you go, and apparently it may have connection to medical fields where pathogens become dormant and then become reactivated.
Okay.
So there is an additional ATP-related factor in there.
Got it. Great. Yeah. Yeah, indeed. What I kind of didn't talk about here is how do you actually exploit the energy gap and use it for stuff.